Flagellin, a protein found in the whip-like flagella that many gut bacteria use for movement, has long been recognized as a potent trigger for the immune system. But only recently has science begun to uncover its more complex and paradoxical role, not just as a microbial red flag that alerts the body to bacterial presence, but also as a molecular communicator in the intricate dialogue between the gut, the immune system, and the brain. At the center of this interaction lies TLR5, toll-like receptor 5, a specialized receptor expressed on cells in the intestinal lining that detects flagellin with extraordinary precision. What makes this relationship fascinating is that flagellin, through TLR5, appears to participate in both immune surveillance and appetite regulation, weaving together inflammation, neural signaling, and energy balance in ways that are now being explored for their relevance to conditions like inflammatory bowel disease, IBD, irritable bowel syndrome, IBS, and even metabolic disorders. In a healthy colon, flagellin's detection by TLR5 activates immune responses that are usually balanced and protective. This signaling helps maintain intestinal barrier integrity and controls the population of motile bacteria, preventing them from invading deeper tissues. But this interaction is not merely a defensive one. TLR5 is also expressed on certain neuropod cells, a unique class of sensory cells in the gut lining that connect directly to the nervous system via the vagus nerve. These cells translate microbial signals, including the presence of flagellin, into neural impulses that influence the brain's perception of satiety, hunger, and well-being. The implication is that some microbes may actually shape feeding behavior through their motility and the presence of flagellin, subtly tipping the brain's decision-making about when to eat or stop eating. Experimental evidence supports this dual function. In animal models, the absence of TLR5 leads to disordered eating patterns and features of metabolic syndrome, including increased appetite, obesity, and insulin resistance. This suggests that TLR5 signaling normally contributes to regulating appetite and energy homeostasis, likely by sending fullness signals in response to certain microbial patterns. At the same time, however, excessive flagellin exposure, especially in a dysbiotic gut with a high number of flagellated pathobionts, can trigger chronic low-grade inflammation. This immune activation may become pathogenic, contributing to gut barrier dysfunction, increased intestinal permeability, and the onset or exacerbation of IBD. In this context, the body's attempt to monitor microbial movement becomes maladaptive, setting off a self-reinforcing cycle of inflammation, dysbiosis, and immune dysregulation. This places flagellin in a curious position. It can serve as both a metabolic signal and an inflammatory trigger, depending on the context. At low levels in a well-regulated microbial ecosystem, it may support appetite control and immune equilibrium. But in disrupted microbiomes, especially those dominated by aggressive or overabundant motile bacteria, flagellin, TLR5 signaling may shift from beneficial to damaging. This duality is not unique in biology, but it offers a compelling explanation for why gut-related symptoms in IBD often include altered appetite, fatigue, and mood disturbances, even before overt inflammation becomes visible on diagnostic imaging. Understanding this neuroimmune crosstalk may open new therapeutic frontiers. If flagellin-driven signaling via TLR5 can influence both feeding behavior and immune tone, then interventions aimed at modulating this pathway could offer benefits on multiple fronts. Selective probiotics that reduce flagellin expression, dietary strategies that support non-motile microbial communities, or targeted blockers that modulate TLR5 sensitivity might help restore balance in patients with chronic inflammation or metabolic dysfunction. Moreover, neuromodulatory strategies that strengthen vagus nerve function could help the body respond to microbial signals more effectively, reducing both appetite dysregulation and inflammatory reactivity. This line of research challenges the artificial divide between metabolism and immunity, showing that molecules like flagellin sit at the intersection of two essential biological systems. What we're discovering is that the colon is not just a site of digestion or immune defense, it's also an information hub, where microbial behaviors are sensed, interpreted, and translated into whole-body signals that shape how we eat, how we defend ourselves, and even how we feel. By decoding the messages carried by molecules like flagellin, We are beginning to understand the gut not just as an organ, but as an active participant in the brain's decisions and the immune system's posture. As we learn more about the gut's sensory capabilities, especially through receptors like TLR5 and cells like neuropods, psychiatry, gastroenterology, and endocrinology may converge in new and unexpected ways. Flagellin may seem like a simple structural protein of bacteria, but in the language of the gut-brain immune axis, it speaks with surprising eloquence.